Hi, welcome to Biology classes with Lakshmi Chandra. We were dealing with genetic disorders in the previous sessions. In that we completed regarding hemophilia. Let's move on to this session towards a new genetic disorder, that is sickle-cell anemia. Sickle-cell anemia. I know that you are familiar with the terms sickle. You all know what a cell is and you all know about the disease anemia. But when these three are brought together, the meaning is entirely different. It is a genetic disorder about which we are saying. Let's see. What is anemia? Anemia is a disease. What kind of disease is that? It's a deficiency disease. You can recollect, isn't it? So deficiency means it is caused due to the deficiency or a low content of something in your body. What is that low content in your body? It is a mineral. And what is that mineral? It is iron. So anemia is caused due to deficiency of iron in human body. So when there is deficiency of iron in human body, what actually happens is we feel tired, we feel fatigued, isn't it? Why so? Because the iron that forms part of our red blood cell and part of the red blood cell called as hemoglobin. Hemoglobin carries oxygen inside our body. So hemoglobin is made up of two parts, heme and globin, where that heme represents iron. So let me just write it on the board. You have red blood cell. The main function of red blood cell in our body is to carry oxygen, and the protein in the red blood cell which is doing that is hemoglobin. Hemoglobin is the protein present in red blood cell which carries oxygen. This hemoglobin, you can just say it is made up of two parts, heme and globin. Heme is the iron part. I am writing the symbol Fe for iron. And globin is the protein part. When there is deficiency of iron in human body, that is when iron, the amount of iron in this hemoglobin gets reduced, the capacity of this red blood cell to carry oxygen gets reduced. So you won't get much energy in the body and you feel tired, fatigued. That disease is anemia. Now then, what is sickle cell anemia? Sickle cell anemia is not a deficiency disease, but it is a genetic disorder. So it is not related to iron here. It is related to a gene. And so when it is related to gene, naturally it will be related to protein. Isn't it? So sickle cell anemia is related to this part of hemoglobin, the protein part of hemoglobin. So let's understand regarding the protein part of hemoglobin in nutshell so that you can understand about the disease in detail. This protein part of hemoglobin is actually made up of two different proteins. Two different proteins. Instead of protein, we can now use the term polypeptides. Because now we are in standard 12. You might have learned in standard 11 regarding polypeptides, isn't it? Peptide is a bond present between amino acids. And when amino acids are linked, you get a polypeptide. When polypeptides are arranged in a particular, folded in a particular manner so that they become functional, you call it as protein. Okay. So this protein, that is globin protein, is made up of actually 4 polypeptides. 2 types of polypeptides, but they are 4 in number. That means the 2 types of polypeptides are alpha polypeptides and beta polypeptides. So this globin part, this globin protein of hemoglobin is made up of two types of polypeptides, alpha and beta. Again, this alpha polypeptide is 2 in number. There are 2 alpha polypeptides and there are 2 beta polypeptides. So totally there are 4 polypeptides which are arranged in a manner so that it becomes functional, and then you call that protein as globin protein, which is bound to iron, and that forms hemoglobin. So globin is actually a class of proteins. I think you have heard about immunoglobulins. That is antibodies present in our body. That is also another globin, that is also another kind of protein. Here in this globin protein, it is made up of 2 alpha polypeptides and 2 beta polypeptides. So they are polypeptides or they are other proteins. Okay. Now, we have learned in previous sessions that every gene has got 2 different forms. Those forms are called as, alleles. Okay, and every gene is responsible for production of a protein. If one protein is not produced in our body, that means that particular gene is not functioning in our body. Or that particular gene may be recessive in nature. So one gene is connected to one protein. Here, since this globin protein of hemoglobin is made up of 2 alpha and 2 beta proteins, there are several genes involved in the production of this hemoglobin protein. It is not just one particular gene. There are several genes involved in that. Now, in this case of sickle cell anemia, we focus on a gene which is responsible for the production of beta polypeptide. So, our focus is on this beta polypeptide. There is a gene which is responsible for the production of beta polypeptide. And you know, this beta polypeptide is made up of 146 amino acids. There is not 1, 2, 3, but 146 amino acids are linked by which bond? Peptide bond. Always amino acids are linked by peptide bond. So there are 146 amino acids which are linked by peptide bonds, and then you get the beta chain. So this is produced by different genes. Now where is that gene present? That gene is present on a chromosome. On which chromosome? On chromosome number 11. So that's how we are going. Okay. So we are telling sickle cell anemia is caused due to a defect in the beta polypeptide region because there is a defect in the gene that is producing it. Okay. And where is that gene present? It is present in chromosome number 11. So chromosome number 11, is it an autosome or a sex chromosome? It is an autosome, isn't it? Sex chromosomes are X and Y. Rest all chromosomes are autosomes. So chromosome number 11 is also an autosome. So this particular disease is autosomal disease, autosome linked. And this is a recessive disorder. Recessive disorder means, I told you there is a gene responsible. Gene is having 2 alleles. If only both those 2 alleles are affected, the person will get the disease. This concept was explained in detail in the previous session when I explained regarding hemophilia. You can subscribe the channel and you can go to the playlist and you can just click onto the playlist so that you get videos in detail. One after the other, it will be played. You will get things as a story and you can just recollect it, understand that. If you can have doubts, you can come. You can ask through the channel so we can have interactions. Fine. Now see, autosome linked recessive disorder. So this particular disease is linked with autosome. So that means it is present on a chromosome number 11. This chromosome number 11 has a gene. This gene is denoted as HB. This gene has got 2 forms, that is alleles. So when you write normal form, you write HBA, HBA. 2 alleles. A here represents they are normal, HbA. Hb is related to hemoglobin. So there is a gene in chromosome 11 which is responsible for producing hemoglobin, the beta globin chain of hemoglobin, and if it is normal you represent it as HbA. So this is producing beta globin polypeptide. The beta globin polypeptide has 146 amino acids. This is a normal case. When the person is getting the disease sickle cell anemia, what happens? Among this 146 amino acid, one amino acid is replaced by another amino acid. That is, just one amino acid is getting replaced. Then the person gets this genetic disorder. So look at the severity of that. One amino acid. Which is that amino acid? I told you there are 146 amino acids. So amino acid 1 I am just putting like this. Amino acid 2, 3, 4, 5, 6, 7, like that it is going till 146. They are linked and they form this beta globin polypeptide. Among this 146 amino acids, just one amino acid is getting affected. Which is that amino acid? It is the amino acid on the 6th position. The rest of the amino acids are same. But only in the 6th position the amino acid is getting changed. What is that change? In the 6th position, it's very important. In the 6th position, you have glutamic acid. This is in the case of normal person. But when the person is getting sickle cell anemia, instead of glutamic acid in the 6th position, another amino acid will come named as valine. I hope you are familiar with the 20 amino acids present in human body. Okay, there are 20 amino acids present in human body, and those amino acids are linked by peptide bonds so that you get proteins. Like we have English alphabets, they are linked, we get words, sentences like that, it keeps on expanding. Same way, there are amino acids which are linked to form polypeptides, which are folded in different forms to form different proteins, and proteins function in different ways, and thus our body becomes functional. We have hair protein, keratin, okay, muscle protein, myoglobin, in the blood we have protein, hemoglobin, like that, so many proteins are there. So this is just simple thing. So coming to our topic, beta globin polypeptide has 146 amino acid. Among that, the 6th amino acid alone is getting changed. What is that change? The 6th position, in normal case it is glutamic acid, but when the person is getting the disease, it is replaced by the amino acid valine. Now why does that happen? Why this replacement? Okay, the answer lies with our gene. See here, if the gene is normal, it will produce normal beta globin chain. But when the gene becomes defective, that is when the gene becomes sickle-celled. When the gene becomes sickle-celled, we can write here like this, HBS. One allele is becoming sickle-celled. That is, in this allele, in the 6th position, it will code for valine. But you see this allele, it will code for glutamic acid. So, this is coding for glutamic acid, normal. So, the person will be actually looking normal. But is the person normal genetically? No. The person is having one gene defective. So, we again use the term here. Right, carrier. The person is carrier, or the person is heterozygous. This carrier heterozygous we have learned in hemophilia disease as well. Okay, so in this case you cannot say the person is diseased, but the person is carrier. Then when does the person become diseased? Only if both the alleles are affected. So both HBS, S I am writing you to denote sickle cell. In that case only it will produce a defective polypeptide where instead of 6th position glutamic acid is replaced by valine by both the alleles. Okay, so the person will get the disease. So here only if both the alleles are affected the person will get the disease. That is why it is a recessive disorder. So two things are important here. It is an autosomal disorder because it is linked to chromosome 11 or it is linked to autosome. Okay. And recessive disorder because only if both the alleles of the gene is affected the person will get the disease. Now the question was why this replacement? Glutamic acid is replaced by valine. The whole answer lies with the gene. Let us go back to that portion. Gene. Where is this gene located or what is gene actually? Gene is functional part of DNA. Gene is functional part of DNA. Right? So we can focus our attention towards DNA. In the case of DNA, I am writing like this. DNA is present inside the cell's nucleus. Isn't it? DNA is present inside the cell's nucleus. Okay, DNA is present inside the cell's nucleus. Sitting there inside the nucleus, the DNA sends information to produce particular proteins. So, the information is with the DNA. And production happens in the cytoplasm. Where is DNA? In the nucleus. Where is the production happening? Outside the nucleus in the cytoplasm. But does the DNA go out of this nucleus into the cytoplasm? No, it sits inside the nucleus and gives direction, only information. So there should be somebody to carry this information from the nucleus into the cytoplasm. Isn't it? Who is that somebody that is doing it? It is a messenger molecule. What is that messenger molecule called as? It is our messenger RNA. Like DNA, there is another nucleic acid called as RNA. RNA are of different types. Among that, one type of RNA is messenger RNA. So what is that messenger RNA? What is it doing? It is carrying information from DNA sitting inside the nucleus to the site for protein production. It is a place where protein production is taking place in the cytoplasm. It is carrying the information. And where is actually this protein production taking place in the cytoplasm? It is taking place on ribosomes. So site of protein production is ribosome. Site of information is nucleus. Who is carrying this? Messenger RNA. How does it carry? It is carrying things in the form of code languages. It is carrying things as codes. What kind of codes it is to carry? See how well things are planned inside each cell. Okay, it is carrying message in the form of code. What kind of code? Code of base pairs. So let's understand what that code of base pairs are. That means message is passed on from DNA through the mRNA to the proteins for the production of proteins. So from DNA through mRNA, what is produced? The protein is produced. How the proteins are produced? By linking each amino acids, the proteins are produced. Okay. For each protein to be formed, there are many amino acids required. So amino acids are the building blocks of proteins. So for each amino acid, there is a code present with DNA. And how is this code actually present with DNA? In the form of base pair sequences. 3 base pairs together will code for one amino acid. Another 3 base pairs together will code for another amino acid. Likewise, suppose the code is GAG. It will code for amino acid 1. Code is something else. It will code for amino acid 2. Likewise. Okay. So these codes are present in the DNA. It is decoded by mRNA and sent to the site for protein synthesis. Proteins are prepared there. How? By just bringing these amino acids together as per the code, and they are linked. This is how it happens. Let us see in this case what is happening. In DNA, in the case of normal person, that is person with HBA gene, both the alleles are HBA, in that case the DNA will have many sequences here, many codes here. We are actually focusing on this particular code, which is responsible for the production of glutamic acid, that amino acid. Okay, so that means this particular code in DNA, DNA is double-stranded. So this is one strand that I have drawn, one part only. The other part, how can I draw that? Yes, based on the complementarity, isn't it? G, that is guanine, will pair with which one? Cytosine, C. A, adenine will pair with thymine, T. Again, G will pair with C. So what I have written is, I have made this single-stranded DNA into double-stranded. I have written the 2 strands of DNA. So same way I can write for this also. What is this showing? This is showing sickle cell hemoglobin. Okay, sickle cell hemoglobin. This is normal hemoglobin which is producing glutamic acid in the 6th position. This I have shown the code present with DNA in which it is producing valine in the 6th position. So here, see, I have written only one particular strand. What is that? G, T, G. So what is the difference? Here it is GAG, here it is GTG. So what is the difference? Difference lies only with one particular base. What is that? A. Here it is T here. So naturally the complementary strand, that is the second strand of DNA, is G is pairing with C, T is pairing with A, G is again pairing with C. So here GAG is replaced by GTG, or we can say instead of A, T is coming. So only in one particular nucleotide the change is happening. One particular base only the change is happening. So it's a single base substitution. Single base is substituted. What is that? Adenine substituted by thymine. So since there has happened a change here, accordingly mRNA will change. Accordingly proteins or formed will also change. But here only single change has occurred. So there is only change in single amino acid. Now look here, mRNA. mRNA will be formed complementary to any one of these strands only. So we're taking this CTC. So again C pairing with G, T should pair with A. But I am not writing A. Okay, let me write A. Then C is again pairing with G. Okay. So here mRNA will be produced complementary to only one strand of DNA. That which strand and why and all you will learn in detail in the next chapter, molecular biology chapter. Okay, so here GAG is produced. How it is produced? Complementary to this particular strand. So same way I have to write complementary here also. It is C is pairing with G, A is pairing with T, C is pairing with G. But here, okay, G is pairing with C, GC is a base pair, AT is a base pair, GC is a base pair. That is the base pair we have learned. But here I have made a mistake in writing. Can you identify that? As per base pair rule, A pairs with T with 2 hydrogen bonds, G pairs with C with 3 hydrogen bonds. So as per the base pair rule, this complementary which is produced here is right. But still I am saying I made a mistake here. A gross mistake here. What is that? Can you identify? Yes. The basic difference between DNA and RNA is what? There is no thymine in RNA. Here I am writing with respect to RNA. There is no thymine in RNA. Instead of thymine, RNA is having what? Uracil. So can I write here thymine? No. Instead of that, what should I write here? I should write here uracil. Now it is correct. Okay, because I am mentioning here not DNA but RNA. So I should not write here thymine. Instead of thymine, I should write uracil. So adenine will pair with uracil here, not with thymine. So now I have corrected it. Okay. Likewise, why I am putting here dots means there are many base pairs. There are many bases on either sides because they have to code for how many amino acids? 146 amino acids. Among that I have chosen only that amino acid. Among that I have chosen only the base which is responsible for the amino acid in the 6th position. That's why these dots are there on either sides. Okay. So here this GAG will code for the amino acid glutamic acid. So normal HB is produced. But see here, it is not GAG which is coming. What is coming here? GUG. So it will not code for glutamic acid. Instead, it is coding for another amino acid called as valine. So this is the molecular reason behind sickle cell anemia. What is the molecular reason? Simple. In DNA, instead of GAG, it is replaced by GTG. Or in mRNA, it is carried, message is carried as GAG. And that is normal, GAG. GUG, GUG, that is abnormal. The GAG becomes GUG. Normal cell will become sickle shaped. So now the term sickle cell anemia. Okay. See here in glutamic acid, when it is there in the 6th position and all our amino acids are correct, our hemoglobin will have a biconcave shape, normal shape. This I am just drawing like this. Biconcave shape. This is not biconcave shape, it is a 3-dimensional shape. So it is having a biconcave shape, normal. But when it is valine, what happens is the oxygen retention capacity of this gets reduced very much. And so the cell which ought to be like this will gain an inward pull. This particular portion will be pulled inward. This portion will be pulled inward and so it will have an appearance like this. So this portion is pulled inward, down, deep like this. So this particular portion is having like this, half moon or something like that. It becomes sickle shaped. Instead of biconcave, it becomes sickle shaped. So what happened? The oxygen carrying capacity, this much amount is destined to carry oxygen. From that the area to carry oxygen got reduced. So the person became anemic. Why? Biconcave shape became sickle shaped. That is why sickle cell anemia. Anemia means there is oxygen deficiency in the body. Why oxygen deficiency? Because the shape changed from biconcave to sickle. Why the shape changed? Because glutamic acid is replaced by valine. Why glutamic acid or how it got replaced? Because mRNA formed is defective. It came with a different information. Why mRNA came with a different information? Because the gene responsible for it gave a defective codon. So the gene is mutated here. So single gene mutation, this is single gene mutation or single base pair substitution, or this is example for point mutation. point mutation. Only single base substitution is taking place here. So I hope reasons for this is clear now for you. So if in the question paper it is asked, what is the molecular reason behind sickle cell anemia? What will you write? Substitution. Molecular reason is substitution of glutamic acid by valine, where in the 6th position of beta globin chain of hemoglobin molecule. Why? Due to single base substitution of the beta globin gene. This is beta globin gene, single base got substituted. So glutamic acid is replaced by valine in the beta globin chain of hemoglobin, and so the biconcave shape of RBC became sickle shaped. The oxygen carrying amount capacity reduced, person became anemic, person is having sickle cell anemia. So here the concepts to be kept in mind is sickle cell anemia is an autosome linked recessive disorder. Autosome, it is present in chromosome number 11. Recessive, only if both the alleles are having the defective gene, the person will have the disease. And molecular reason is substitution of glutamic acid by valine in the 6th position of beta globin chain of hemoglobin due to single base substitution of the gene responsible for production of beta globin gene. So with this we wind up for today's topic like sickle cell anemia. Next day we will come with another genetic disorder, phenylketonuria. Wait till that session. Bye for now.